Molecular basis of inheritance. After Mendel's work, it took long time to realize that DNA and RNA are the genetic material, but in majority of organisms, it is found that DNA is the genetic material, whereas in case of certain viruses, RNA is the genetic material. Otherwise, RNA usually functions as messenger, and there are few additional roles of RNA, that is, it works as adapter molecule as well as it shows catalytic properties in certain cases. In this chapter, we will be dealing with structure of DNA, the replication of DNA, transcription process, that is how RNA is made from DNA, and translation, that is protein synthesis. Let's start with the DNA. As you know, DNA is deoxyribonucleic acid. It is long polymer of deoxyribonucleotides, and length of DNA is equal to number of nucleotides present in it. This length is specific to organism, that is, this length of DNA varies from one organism to the other organism. Example, in case of bacteriophage Psi 174, this is the name of the strain Psi 174. It is found that the length is equal to 5386 nucleotides, whereas in case of bacteriophage lambda, it is found that length of the number of nucleotides is equal to 48502. Can you find out what is the number of nucleotides present in human DNA? Human DNA is found to be 3.3 into 10 to the power 9 base pairs. Structure of polynucleotide chain. The nucleotide consists of three components: nitrogenous base, pentose sugar, and phosphate group. These nitrogenous bases are further of two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidine are thymine, uracil, and cytosine. In cases of RNA, thymine is found to be replaced by uracil. Now, this nitrogenous base is combined to sugar with N-glycosidic linkage, and this gives us nucleoside. On this nucleoside, fifth position of OH, when phosphate group is attached through phosphoester bond, it gives us nucleotide. This nucleotide is attached to another nucleotide with phosphodiester bond, and this gives us dinucleotide. Now, after this dinucleotide, the polymer is, because of this linkage, the polymer is found out that at the 5 end of the sugar, a free phosphate group is always found, and at the 3 end of sugar, OH group is always found. In case of RNA, there is an additional OH group at every second position. Now, M. Wilkins and R. Franklin were the people who gave us double helix structure of DNA. This structure says that the base pairs are complementary in nature. That is, if the sequence of base pair of one strand is known, the base pair sequence of other strand can be guessed because they are complementary in nature. Usually it is found that adenine is paired with thymine and guanine is paired with cytosine. Both strands, they act for template of synthesis. That is, if this is the double strand DNA structure, both the strands will act as synthesis for the new strands, and thus the new double helix structure so formed will be identical to the parent DNA. This clears the reason why DNA is the genetic material.